Thank you all for coming back. We have our first talk here by Julian, who will introduce himself. Thank you.
Good afternoon, everyone. I hope you can hear me all right. My name is Julian Lilkendey, and I'm from Auckland University of Technology. Today, I want to make a simple argument that how a fish behaves moment to moment is one of the most sensitive signals we have of a reef under stress, and that we can now read that signal automatically in 3 dimensions without a diver scoring a single behavior by hand. By segmenting its behavioral trajectories. So for decades now, behavior has been our early warning system. When a reef is disturbed, fish change, what they do long before we see it actually manifesting in biomass or survival. But behavioral ecology on reefs has a bottleneck, as some of you might know. Scoring behavior is slow, subjective, and tedious. Our students, watching video, ticking boxes, very, very tedious to do. And the metrics we get out of it are rarely comparable across species or across studies. This is what I encountered with this study. So behavior, our most sensitive indicator, has stayed our least scalable one. More mobile reef fishes make it even harder. They are distributed foragers in a structurally complex, three-dimensional, constantly shifting habitat. Their energy budget isn't set by swimming alone, it's set by the fine-scale costs of searching, biting, staying vigilant, all against a backdrop of environmental constraints. So let me anchor the concept real quick and remind my colleagues of what we actually mean when we are saying a reef is functioning. Belwood and colleagues, Robert, hi there, made a point that on reefs, the word function is everywhere, but almost never defined. Most studies measure an easy trait as a proxy and assume it maps onto function. Their fix is a single universal definition. A function is movement or storage of energy or material. And this should hold true from cell to the global level. Here's the part that matters, for me at least. Functionality runs across 3 layers: morphology, physiology, and behavior. Behavior is where energy is actually spent, and it's the least quantified. That's the gap I like to fill. The framework for filling it is what I call the energy seascape. It grows out of the energy landscape concept. So movement is costly. That cost varies across space, and animals settle where foraging is cheapest. Arostegui and colleagues extended this concept to the ocean into 3 dimensions. So for an open ocean predator, the cost of foraging shifts by as much as a quarter, depending on environmental factors in the seascape. For coral reefs, I propose we build the same thing, a 3-dimensional field of how costly it is for fish to live, forage, and stay in energy balance. Reconstructing from field metabolic rate, behavioral state, and the environmental drivers around it. So not just where fish move, but what it costs them to be there. To test this, I went somewhere the cost of living on a reef is visibly changing. AUT's Amagan Sabetian invited me to tag along with him to Kolombangara Island in the Solomon Islands. Kolombangara means water sheaf. A near perfect cone of volcano fringed by coral reef. Around 80 rivers carrying rainwater from the summit down to those reefs. And after decades of logging, they carry sediments with them. The result is a steep turbidity gradient running along the coast. Natural experiment in environmental stress, if you will. So parts of this natural turbid reef have now transformed into what I would call an anthropogenic turbid reef. The rest of the fringing reef, at least back in 2023, still looked quite healthy. And of course, the matter is not abstract. For the people of Vavanga, the reef is the center of their daily life. Food, income, and culture. So you can imagine that if the energy economy of the reef shifts, their economy will shift with it. So that's at stake here. Of course, it's a beautiful place to work in. So how do we measure the cost of behavior in the wild without even touching fish? We used stereo video, so 2 calibrated video cameras. You feed the system points of known distance and it reconstructs the scene in 3 dimensions. Here is me calibrating somewhat awkwardly back in Eilat in 2018. And here's my former master student Emily doing it far more gracefully. Her second name is Grace, in the Solomon Islands. From this, we could reconstruct the full 3-dimensional trajectory of individual fish. This here is a triangle butterfly fish, a species so tied to coral reefs that it's a living indicator of reef health. Speaking of bottlenecks, 3 students worked a combined 2 years tracking 269 fish, totaling almost 30,000 XYZ points. That means they clicked and combined 60,000 times, once in the left and once in the right camera. And here on this drone footage, you can clearly see the turbidity gradient ranging from the north to the south. And this is accompanied by also a substrate gradient ranging from a lot of dead coral in the north to fewer in the south, and higher epiliptic algae metrics in the south as well. So we do see a turbidity gradient as if realized by Forer Ule and visibility in the system, and dead corals and EAM, and also in live coral. From each trajectory, we were able to derive overall dynamic body acceleration, the sum of acceleration across all 3 axes. It's a validated proxy for how hard animals are working, and already the pattern is striking. If you have a look at triangle butterfly fish here, ODBA is highest at Uvuku and lowest at Church. This is Nina Chittekatte's work, and I draw heavily from it. We translated this movement into field metabolic rate, calibrated against physiological trials, so data from literature, turning acceleration into energy. And what we see here is significant differences in the energy expenditure in reef fish across the reef gradient. We examined 5 taxa in total. Correcting for body sizes and species, we recover a movement energy seascape across the sides. So this already pretty much looks like what I promised, a real measured version of that cost surface. But energy alone doesn't tell you what the fish is doing. So we extended a concept called behavioral change point analysis into 3 dimensions. This methodology has originally been developed to track birds via GPS. And so we elevated into 3 dimensions with speed and turning angle to detect the moments a fish switches behavior and cut each track into bouts. And now we come to the unsupervised approach. We then let k-means clustering sort these stages into hovering, area-restricted foraging, and direct transit based on the velocity of the fish. Clustering looks like this. One consistent scheme applied identical across all sites and taxa. That's what I would call scalable. This is a behavioral budget for all 5 taxa broken out by site along the turbidity gradient. Each bar is a share of track time a species spent hovering in area-restricted foraging or in direct transit. Every taxon has a distinct signature. Butterfly fish, for example, live in hovering and area-restricted foraging with almost no transit. Parrotfish, here Chlorurus blackeri, is transit-dominated everywhere, true roving grazer. And several species even swing into direct transect on the most turbid sites in the north. The perks of working with video is that you can actually ground truth your unsupervised learning approach. So let's see what the fish were actually feeding on. The triangle butterfly fish was feeding on acropora, so a real specialist. The same with Stenochaetus striatus, the surgeonfish, who completely stopped feeding on EAM in the most turbid side. I guess Sterling Tebbit would approve this method. And yeah, Vagabundus turned out to be a true generalist. So these specialists, they are partitioning the reef depending on its substrate. So we uncovered 2 signatures of stress across the reef. The 3 dietary specialists, defined from Manley's feeding ratios, are the ones affected by the gradient, but in different currencies, while the generalist is buffered. The effort escalator, Baronessa, keeps the same behavioral mode but pays more energy. Lunulatus, Orchidodum lunulatus, and the surgeonfish are what I would call mode switchers. So they have flexible behavior, swinging into direct transit at turbid uvuku through their net energetic change, which is not significant. And we have the non-responders, so generalists who not changing, the 2 species in the middle there, who are not changing their behavioral mode and also don't face an energetic penalty. So you mentioned several bottlenecks there. Also recording, right? Storing the behavior, and in our case, also recording XYZ coordinates in fish. How am I planning to overcome this? And so we've developed this AI-driven fish tracking algorithm, which can track multiple fish at the same time. And we are now in the process of adapting it for the Solomon Islands coral reef. So this is how I hope to overcome the scoring bottleneck in the future. So let me pull it all together. By reconstructing behavior in 3 dimensions and segmenting it automatically, we got a species comparable observer independent readout of what fish are doing and what it costs them. The energy seascape measured. And it responds along the turbidity gradient. The same species works harder, forages more expensively, and shifts its behavioral budget at the individual scale. The layer where these responses appear first, and one that community level metrics aren't designed to capture. So before I finish, this work only exists because of a partnership with Bavanga community, and Amagan will bring our results back to them to inform them about the state of their coral reefs. And in this effort, we hope that we measured what really matters and hopefully advance local coral reef management for future generations to come. I thank my co-authors, and particularly Amagan Sabetian. I'm really proud of this moment, so thank you very much.